Hopefully it has started recording. So Bio 208, this is our lab over blood. We have kind of three things we need to go over. We need to go over hematocrit, we need to go over blood typing, and then we need to go over the blood boards and the cells that you need to be able to identify by picture, by model, or by slide, okay? But we're not gonna do computers, we're not gonna do microscopes through the computer, so you would have to do them by picture, okay? So the big thing we start with is that you all, at the very beginning of the chapter, remember that you get a vial, all right? And in that vial, there's blood, right? And it looks like that, okay? And so one of the key things we have to do to figure out is, as a connective tissue, what part of this fluid is going to be, again, just that, is going to be the water and maybe the solutes and the glucose and the proteins, all right, and the phosphates and the small molecules. And then what part is going to be all the cells? And then for good measure, every now and then, one of those white blood cells, okay? And so one of the first things we do with whole blood is we centrifuge it. And by centrifuging it, you can spin and everything is going to, like in the scrambler ride, everything moves to the outside based on weight. So the heaviest things are going to drop at the bottom, right? And then this imaginary line where we see the fluidy part and then the real thick formed element part, that line, we tend to call it the hematocrit. Okay? And we make some leaps and jumps, and we say the hematocrit kind of represents how much of the blood is red blood cells, because most of this is red blood cells. And then we make another leap, and we say because most of it is red blood cells, how much of the blood is carrying oxygen to the tissues. Okay? And so again, 37% is kind of like, we don't want to go below that. If we're below that, we're going to throw a word around called anemia. All right? Above 55%, we start to think bad things like leukemias or too many red blood cells, and you throw around a bunch of words related to cancer, maybe. Okay, because you're overproducing them, or you're super dehydrated. Okay, so 55 to 37 is kind of that range where we want the plasma versus the formed elements. And we call them the formed elements because remember, you can kind of make an argument a red blood cell is actually dead. It has no nucleus, it can't respond to its environment, it cannot divide, it cannot multiply. And a platelet's kind of dead. It's not even a true cell anymore because it's just this cell piece that has some organelles, but it doesn't have a true nucleus, it cannot divide, it cannot multiply, and it's on a finite window of death. Okay, so over here behind Magdalene and Olivia, all right, is a little kit. And I'll walk over, well, I'm not gonna walk over there right now, but I'll walk over there later and I'll try to grab them. I did some blood samples and they're labeled A, B, C, D, and they're just some dye and some goo and they're suspended, but then when you spin them, the goo is supposed to fall down. There's a lot of operator error in that experiment. Jim can probably remember that if you don't do it right, you get it all over yourself, and if you don't shake it really good, when you pull up the fluid, you're really just pulling up the fluid, not the cells. When I was pregnant, I would go to the doctor in Virginia because that's where I was living at the time, and they would prick my finger almost every appointment, and they would spin my blood and look at my hematocrit. And part of the reason why they were doing that, as pregnant women, when you're starting to feed oxygen to not just you and the baby, we want to make sure that you're not getting anemic and not getting low red blood cell count as your fluid is expanding. So that way we know that you're feeding yourself and you're feeding a growing entity that needs a lot of oxygen, enough oxygen to have normal function. And so when you're pregnant, chances are, because your fluid expands, and so your red blood cells go from being in a gallon of milk to like a gallon and a half of milk, you want to make sure you're giving your body those proteins, that folic acid, that iron, everything it needs, so as it makes more red blood cells, it can fill them full of hemoglobin. Okay? All right. Next part is the little kit in front of you. Okay? And everybody got this, so I can erase this, because I think you'll see better. Can y'all see me okay over there?
Yes.
Okay.
Yes.
So, in red blood cells, at one point in time, you had some DNA, okay? And on the DNA, there's a gene, and that gene might be for the A antigen protein. And if this red blood cell, before it was a mature red blood cell, made a little message RNA of that DNA and then made a little protein, they could put that little protein on its cell membrane. And that protein on the cell membrane is part of how we know the cell genetically belongs to us, because it's making proteins that are part of our genetic code. That protein is also one that's capable of having an antibody made against. So anything that's put on the cell membrane that could potentially have antibodies made against them are known as antigens. And so this is our A antigen, and then this is an antibody, and it's usually shaped as a Y, and this part right here and this part right here are kind of like puzzle pieces that want to meet up with this part of the DNA or this part of the protein. And so like the amino acids here, you know, here there's a lot of like lysine, tryptophan, tyrosine. Well, then on the antibody, this area is going to have like probably the mirror of the amino acids that want to like merge and form puzzle pieces and bind to those guys. Does that make sense? This is why you have to take chemistry, so you understand this crap. Okay? All right. So this antibody is known as the anti-A antigen antibody, and that's a lot of A words, right? And in physiology, we like to shorthand everything, so we just call it the anti-A antibody. Okay? All right. For some of us, you have DNA that doesn't have the A gene in it. It has a different gene that is going to produce the B antigen. And so it's going to make mRNA, and then it's going to make proteins that are going to, again, embed on the membrane of the cell, and they're known as B antigens. And same thing, there's going to be an antibody made, and this region of the antibody is going to want to bind with the amino acids that are exposed and present on the outside of the red blood cell for the B antigen. Yes?
What happens during pregnancy if the mother has A antigens and the baby has B, for example?
Well,
or if they...
we'll get there, but let's just worry about this right now.
Okay.
Okay? All right? And so our third option would be you have DNA, and at one area it has the B, and at one area it has the A, and so it makes A, and it makes B antigens on the cell. And then there are other people, and they have DNA, but it doesn't have any of these genes. Okay? And so in our little well, all right, if I take Jen's blood, and I take Jen's blood and I put it in my wells, all right, so Jen, wave your hand in the back so everybody knows who you are. All right, we're going to exclude this guy right now. On your little well, it has, this is A and this is B. Now remember, your DNA is determining if you have these proteins on the membrane or not. And remember, I know what these proteins look like. So again, good chemistry. I can make an antibody in my chemical lab that if it finds this antigen, this protein, it'll bind to it, right? So in our little Petri dish, I am going to add some antibodies to this well that if there's a antigen on Jen's cells, it should find it and bind it. If here, I'm going to add to this cell some B antibody, anti-B antibodies, and if she has B antigen, I'm going to find it. If she has both, they'll both show it, right? And if she has neither of those antigens, the antibodies won't find anything. Okay, so this is how we come up initially with the A blood type, the B blood type, the AB blood type, and the O blood type. All right, now, different place on the DNA, different site, different genes, because you know you have 46 chromosomes, right? You got a lot of stuff in there. So in a different DNA piece, you have potentially another gene known as the Rh factor. Sometimes it's even called the D, because I guess they tried to keep the AB thing going. The D antigen. And it can make a little protein and stick it on the cells if it's present. Okay? And again, because it's an antigen, by definition, I can make antibody to it. So over here in Jen's blood, I'm going to put some antibody to what's known as the Rh factor. If it binds and finds the Rh factor, I know Jen's DNA has the Rh protein. And I would then say she's positive if it's there, or I would say it's negative. So blood typing is basically testing to see, genetically speaking, do you carry a gene for the A antigen? Do you carry a protein known as the B antigen in your genetic code? Do you carry both? Do you carry neither? That's one part. Then the plus or minus tells you, do you carry the positive or the negative? Now, going into pregnancy, usually mom will not abort a baby if they are different blood types. And again, it goes back to chances are, most of the time, baby and mom are pretty similar because there's a 50-50 chance, unless dad, if it's good old Mendelian, if mom's A positive, there's a chance, there's like 3 or half a chance the baby's going to be A, and then, only a 25% chance baby would be completely something different. But usually the baby and the placenta are not. The placenta doesn't let the plasma proteins usually, I guess, supposedly cross, so baby should be okay for that purpose, or the placenta is protecting it. Now, for the Rh factor, the placenta might not protect baby from the antibodies. And so the Rh factor, if mom is negative, the first time she has a pregnancy and baby ends up being positive Rh factor, at certain points in time during the birthing process, mom could be introduced to the Rh factor blood and could make antibodies against it. And so then in that next pregnancy, it's possible that she could try to kill the parasite because that's what she sees the baby as. And so back in, I guess, medieval times, you had one baby and it was a girl, and then you never got pregnant again, never had another kid, and you got your head chopped off. It could have been maybe they had different Rh factors. Speculation, merely speculation, okay? But now, if we know mom's negative and we know dad's positive and we know the baby might be a positive Rh factor, they give antibodies to the antibodies. And so they give a big shot. So these antibodies are then attacked by antibodies to destroy them. So that way mom would take them into the, usually the second or third trimester and not stillborn or lose the baby. I've never heard and seen anything that says there's a problem with the different A, B, or AB antigen presentation. The only time in pregnancy, and they have a thing in your book that talks about this, related to the Rh factor, but I don't think the blood typing is an issue. But it's a good question because in theory, Again, mom has a 25% chance of having a baby that might not look like her, but maybe first baby does look like her. So then it's maybe the second or the third baby that before she gets exposed to something that might be foreign. And again, the only time you make antibodies to something is if it's exposed, the blood mixes. And usually the way the placenta works is the blood doesn't mix. They come close to each other, but they're not mixing. Okay. Any other questions? Okay, so in your little kits, you should have, and I gave all the kits away, so I'm going to turn y'all around, and y'all are going to watch. You should be able to get one of the little trays, okay, and pick out one of the little plastic trays, okay? And then you should have 2 bottles of Mr. Jones, Mr. Green, Mrs. Smith, okay? So what you're going to take is take the red blood, your fake blood of whoever you have, and put 3 to 4 drops of blood into your little plastic tray. Put 3 or 4 drops in the A well, put 3 or 4 drops in the B well. You just need one of those little plastic things, just one. All right, so put 3 or 4 drops. You can put a little bit more. You want enough that when we add the 2 together, there's enough, because we got the dregs, we got the bottom of this has been used in 111, and we get the leftovers. So put a few drops in each well. So same blood type, same one. No, you keep using the same one. Put them in all 3 wells. So put 4 or 5 drops of red blood in each well. Okay, so imagine you just pricked your finger and you put 4 to 5 drops of your blood in each of the 3 wells. Okay, now take the blue top is anti-A. So what we're doing is we're saying, here is antibody that is going to find the A antigen. And if they find it, I know genetically speaking, there's A antigen here. Okay, so drop in 4 or 5 drops of that. Make sure you put it just in the A well and it doesn't spill over. Okay, and don't tap the top into the blood because then you're contaminating it. How many drops? 4 or 5. You know, you want to get a good enough sample that hopefully there's some goo to mix. Okay, then do the same thing with the yellow. The yellow is the B, anti-B. And if it looks like you need more, add 2 or 3 drops more blood, 2 or 3 drops more of the blue, and same thing with the yellow. And then do the green in the RH well. Okay, now this is all chemistry and this is all fake blood. So, in a real blood test, remember that picture in your slide, how everything aggregated really quickly, the red cells to the like a bead in the center, and everything that was left was kind of the plasma around it? If this is real blood, it looks like that. This is not real blood. So, it's going to take a few minutes, and then in a few minutes, if you have antibody and antigen interaction, the A well and the RH well, it's going to look like some ice crystals form. So it's going to look like almost like hair is sitting on the top of the fluid. The B, if it's positive, should turn a slightly different color, and you should see what looks like some little beading at the bottom. Okay? And you have to give it a few minutes. So, and some of you have Mr. Jones and some of you have Mr. Green. And again, the kit is for a little lab to find who's the baby's daddy. And so they do it in the genetics lab where everything's by Mendelian genetics. And they try to figure out Mrs. Jones has had a baby and the baby is blah, 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 and who's the baby daddy? And they work backwards of here's what Mrs. Jones's blood type is. Here's Mr. Green. Here's Mr. Brown. Here's Mr. So-and-so. And if the baby is B positive and mom's A, then the daddy has to be something with the B, right? Because that's the only way the baby would be the B. Okay, so it's going to take a few minutes to do that. All right. Did y'all see? Okay, what's going on?
Yes.
Okay, so generally on my lab exam, I will ask 2 of these questions. So I'll have 2 of these stations set up because a lot of the lab's going to be online for the people online. There's a good chance I'll do it by pictures. I'll take one of those like blood typing wells and I'll white out all the answers, and then I'll point to what is this well person's blood type. So being able to read these is important. If we're able to take labs at week 6, if we're not virtual by week 6 because this place hasn't shut down, expect 1 to 2 of these stations where you'll walk up and read the kit. Okay. Does anybody see anything happening yet?
What did you say the different ones would look like again?
So the A well looks like glass forms on it. It almost looks like ice forms on it, or a piece of hair or string is sitting on top. And then the B will change a slightly little orangey color.
I think that's the B.
Yeah,
It's changing.
and it'll, I don't know.
Yeah, I don't think it's gonna be possible.
It's probably hard to see online, but
Is this second one the B?
so A, B, and then RH.
Okay.
Okay. Oh, we're running out of time. Okay, so let me get to the last thing before you guys get kicked out. And how did you guys think this went so far?
I think it went good.
Okay. So the last thing we got to talk about is the blood board. Okay. There's a picture of this in your lab handout, correct? Get him out of the way. Okay. All right, he's throwing orders right now. Okay, so the blood board. So again, you don't necessarily have slides. So the blood board is one way I can put stickies on and ask you things as well as pictures. Okay, so if I was to take a droplet of blood and put it on a glass slide and put a top on it, this is what you would see. You wouldn't see this exactly, but similar to this. So the first thing you notice is this yellow background represents our plasma, right?
Okay.
And you can't see sodium, potassium, and glucose, and creatine, and LDLs, and triglycerides, but they're in there. That's what's suspended. Even a little bilirubin, a biliverdin, a little bit of other binding proteins, hormones, they're in this yellow background. What you do see is a lot of these red things, right? These are the red blood cells, and then the blue things are the platelets, okay? All the other cells, if we did this right, if we had 100 cells on here, how many of them would potentially be something not red blood cell?
One.
Because 99% of what we get should be red blood cells. 1 to less than 1 would be a white blood cell. So this is a lot of artistic liberty going on here. But for the white blood cells, the most numerous are these that are polymorphonuclear. They have granules, and we talked about that yesterday in the lecture. Their cytoplasm stains a little bit of a lighter background, but they're rough looking because of the granules. That's all the vesicles full of chemicals. And polymorphonuclear. So they make their nucleus into these multiple bead looking things. This guy is, again, spits out its vesicles, spits on a cell first, and then tries to eat pieces and parts of it. Some of the vesicles travel and recruit more neutrophils. And since so many of them are in the blood, they are part of what the cytokines are like ants. They're going towards the bloodstream and helping to pull these guys out of the bloodstream to come help in an injury site. Some of those cytokines will even get in the bloodstream and head to bone marrow, because what do I need to do if I just use the 1,000 neutrophils to clean up an injury? I need to replace them. So I need to signal to my hematopoietic stem cell, and some of those cytokines are like interleukins, maybe some colony stimulating factor that can then start to trigger hematopoietic stem cell divide, become a myeloid cell, become what will eventually become a neutrophil. Okay, the other one is, you can't see it, is this eosinophil, and there's one right here. It's going to have vesicles like the neutrophil because it spits first and then eats. The vesicles stain a little bit different in color because remember, these guys, they're trying to, if this guy's going to attack like fungus, this guy is going to attack like viruses. Like they're trying to cover their bases. I'm going to throw acid, you throw base. And one of us will kill it and we'll all eat the pieces, okay? And they're binuclear, all right? So they make 2 lobes. These are eosinophils. And remember, from chapter 4, together, these 2 guys were known as your microphages, okay? This big guy, the biggest cell, is your monocyte in circulation, but when it goes and takes up residence in the spleen, in the liver, in the lymph nodes, in your skin, in the connective tissue behind your and around your heart, in the superficial fascia, in your wherever you have connective tissue, which is almost everywhere, okay? It's known as a macrophage, and its mojo, it has vesicles, not as many. It eats your cell or your pathogen first, and then digests it with the lysosome help. And then it goes and presents it to the T cell and says, hey, I ate this. Is it self? Is it good? Did I do good? You know, it's like a little kid. Did I do good? Did I ate it? I ate it. I ate it. You know, I ate poop, right? Okay. And the lymphocyte says, no, you didn't eat poop. You're fine. Okay. You're fine. All right. This represents a lymphocyte. Remember, you don't know if it's a B cell, a T cell, a memory cell, a suppressor cell, a natural killer cell. You don't know. All you know is that, again, it's a lymphocyte. To be able to tell if it's a T cell, a B cell, or whatever, you would need to know what are some of the proteins on its membrane. And you are not able to do that by the naked eye. So when we take blood now and we drop it into a machine, the machine can shoot lasers and do some analysis really quickly. And it can tell you big picture what cells you have and the numbers. And then it can sometimes even tell you what type of lymphocytes you have. Okay? So those are the white blood cells. So again, by picture, and again, I did that yesterday in lecture, and that real quick, by picture, by model, you would be able to identify your white blood cells, or by the information I give you. This guy is the most numerous. This guy is the biggest. These guys spit first and swallow second. This guy swallows first and breaks down second. These guys are part of specific defenses typically. Can't tell them apart unless I do more invasive molecular tests. All right, questions from the online folks before you go bye-bye.
The blue one with purple, what's that one again?
This is known as a monocyte when it's in circulating blood. Once it becomes outside of that, it becomes a macrophage. Oh, I forgot this guy. This is the third granulocyte. This one is the basophil when it's in circulation. When it's in your connective tissues of your airways, connective tissues of your skin, it's known as a mast, M-A-S-T, mast cell. This is the guy we usually associate with, what do you call it? With asthma attacks and allergens, okay? And this is the guy like Singulair and some of those leukotriene inhibitor. They're trying to inhibit the cytokines that come out of this guy that cause inflammation and white blood cell activation and changes in your blood flow and the reaction that causes your face to swell up and your airway to close down.
Okay.
Okay?
Yep.
All right. So you guys are going to probably go away. Thank you guys for coming. Do you have any other questions?
I don't think so. This will be recorded and then it'll pop up. When will it pop up so I can rewatch it?
When I figure out how to post the recording.
Okay.
And where I can figure out, I'm hoping somebody in here knows how to post a Zoom recording, but I will try. Okay, because it's saved on this computer, so I'll try to get it somehow put on YouTube page or on the Blackboard site, okay?
Okay, thank you.
Thank you guys. So again, next week, if you can come to your normal lab, great, but if not, we'll do this again on Thursday.
I'll be there again on Thursday.
And I'll try to be quicker so we get more of the time spent on the hearts, because it's heart dissection.
Okay.
Okay, so what do you guys have for questions? It's this one. So come on up and grab this if you want, or you can grab that. Okay, let's look at our things real quick. Does anybody see? So yeah, I would probably say there's not a lot in there, but it could be that you have nothing for A and RH, but you might be B negative. What does yours look like? Yeah, who do you have? Mr. Jones, Mr. Green?
I have Mr. Jones.
Mr. Jones? What did you guys come up with? Oh, you guys definitely look like you got A positive. So if everybody wants, put that over there. Come look at what A positive. Oh, you got A. You look like you got A positive too. What did you get? then you would have what? Nothing happened. Oh, what? Oh, negative. Yeah, this one looks like it's a B positive. So she's getting, you're getting crystals in the A. You look like you get those icicles in the B, and then, I mean, in the RH, and then it looks like she's even getting the beading in the B. So this would be, and again, y'all can take pictures. Y'all can have your cameras out if you want. Y'all can take pictures of this. Y'all can take pictures of the board. Although if you do have the board as a picture, what did you get?
B negative, I think.
Yeah, maybe. That one would be, I would be waiting a little bit longer to see if anything happens, but it would either be maybe B or nothing.
Okay.
Okay, so here, this one, somebody started to label. So if anybody wants to take pictures of this one, it is available as well.